Hey everyone, this is a presentation about my project in experimental molecular cell biology, where the goal was to induce cell death by using a random reagent. For my experiment, I added a droplet of energy drink enhancer to a culture of COS7 kidney cells. A key part of this course was also learning how to responsibly use AI for research. At the bottom of this slide, you'll see images generated using prompts I created to mimic my own data. On the left, Adobe Firefly really struggled. It looks more like a cartoon of an aquarium than a microscopy. Night Cafe did much better. If I didn't know, I might have believed this was real. The image on the right is my actual fluorescence data, which I used to create these prompts. First, to find out if my cells died, I took my cell cultures and stained them with nuke blue, annexin-5 and propidone iodide. Nuke blue is a stain that turns the nucleus of cells blue. This lets us know that a cell is present. Annexin-5 has a high affinity for phospholipid phosphatidylserine, which is exposed after apoptosis. Propidone iodide stain will bind to DNA or RNA that has been exposed out the cell membrane, which indicates a cell has undergone necrotic death. The first reagent I used was actually hot tub water from a public gym because it seemed like a funny idea. I treated U251 glioblastoma cells with 25 and 50% concentration of hot tub water. For context, U251 cells are a model of a type of brain cancer. The results of this were pretty gross. There were almost no visible cells left on the slide, but this outcome wasn't very informative because the sample was overwhelmingly noisy. This was probably due to high chemical or microbial toxicity from the water. I learned from this mistake that too harsh or complex of a reagent can make it impossible to interpret the results. It's much better to go with something that has more of a known concentration so you can start isolating components depending on your goals. The second reagent used was the MiO Energy drink enhancer at 15 and 5% concentrations. I chose this reagent because I was curious about the effects of caffeine toxicity, especially when mixed with other synergistic compounds such as guarana, ginseng, and vitamin B3. This had absolutely beautiful results. You can clearly see the green outline of annexin-5 on the membrane of the cells, and nuke blue and propidone iodide are perfectly lined up on the nucleus, showing that this reagent had strong necrotic effects. In conclusion, energy drink enhancers can cause necrosis in COS7 cells, as shown by the clear annexin-5 and propidone iodide stain. This project was a lot of fun and gave me a much greater appreciation for how broadly applicable cell biology techniques are. I had no idea that so many questions could be answered using just immunofluorescence. I've also changed my opinion on AI tools. I came into the course skeptical, especially after AI has fed me misinformation in the past. But I found it very useful for things like automating simple research tasks, such as analyzing an ingredients list. I've started using it in my day-to-day life to check if a skincare product is safe for me. Looking ahead, I'd love to test isolated compounds like caffeine and nicotinamide individually and in combination to better understand their roles in cell viability. Thank you so much for your time and listening to my presentation.